Hello friends, welcome to Quick Glance videos. In the last video we studied about prokaryotic cells. So this video we will have a small explanation about eukaryotic cell structure and functions. I will be going in brief so that we can finish within 10 to 12 minutes. The cell. The cell is the smallest unit of the life that is capable of performing life functions. So the cell can be a bacterial cell, plant stem cell, RBC, WBC, root cells, nerve cells. So they have certain functions to be performed. So they are divided on the basis of functions. All living organisms are made up of one or more cells that may be unicellular or multicellular. All cells form pre-existing cells, and that is through cell division. So they come from the pre-existing or same cell, and that's by cell division. So these are some of the features of cell. Now moving to eukaryotic cell. Eukaryotic cells are cells that contains nucleus and organelles. So nucleus and organelles are present in eukaryotic cell. In prokaryotic cells, these were absent and are enclosed in plasma membrane. This is one of the major differentiator between eukaryotic and prokaryotic cell. Eukaryotic cells contains variety of structures called organelles, which perform various functions within the cell. So it's like if you see any company or organization, so it has a complete structure is there. It has various divisions. So you have a various divisions like you have a HR department, you have a finance department, you have a CEO's office, you have a work area, you have a developer sitting there. So these compartments are divided and they have a different functionality. So the same way in a cell, the functions are divided between organelles and they all perform together for the cell to survive. Eukaryotic cell diameter ranging from 10 micron to 100 microns. We'll discuss about the comparative size of various cells. So if you see in microns, the size of say in the bacterial cell is approximately 1 micron. So as we move towards forward, so 10 micron to 100 microns, we have plant cells, animal cells, human cells are there, and going further we have till 100 millimeter we have cells. So some cells they are seen only by electron microscope, and some cells like prokaryotic bacteria and plant cells light microscopy require, and bigger cells can be seen by naked eye. So these are comparative size. As the size of the cell increases, so various organelles in the cell increases to help the existing organelles. The transportation structures are there to help in movement of materials or nutrients towards the various organelles. So as the cell size increases, the transportation structure also increases in the cell. So we are discussing about plant cell and animal cell. So there are many organelles same in plant and animal cell, but in plant cells some differentiation are there, which we'll discuss in future slides. So this is a structure we are going to discuss in various details. The cell structure and function. The plasma membrane is made up of phospholipid. It regulates the movement of material between the cell and outside environment. It's made up of a phospholipid bilayer with embedded proteins that separates internal content of the cell from its surrounding environment. So this is the structure. So it's made of phospholipid. This is inside the cell. This is outside the cell. So this is this forms a membrane. So in case of plants, the structure of the external structure of plasma membrane, which is also called the cell wall, is much rigid in covering and it protects and provides structural support to the cell. So in plants, the outer structure is much more rigid and it consists of organic molecule called cellulose. So this forms the plasma membrane. So we'll get into details of plasma membrane in later slides. Cytoplasm. Cytoplasm comprises of content of the cell between the plasma membrane and the nuclear envelope. So between plasma membrane, this is the plasma membrane and the nuclear envelope. So this area, the content is called cytoplasm. Made up of organelles suspended in the gel-like cytosol. the cytoskeleton and various chemicals, and 70 to 80% of water. These are the constituent of cytoplasm. Cytoskeleton. Now the cytoskeleton is a network of protein fiber that helps to maintain the shape of the cell. So it's like a skeleton system in a human body. So this is a small structure which helps to maintain the shape, secures the organelles in the specific location. It helps to keep the organelles without floating here and there in a specific location, allows cytoplasm vesicles to move within the cell, enables the universal organs to move independently. So this is the cytoskeleton. So it has three types of structures, it has three types of fibers it has, and the actin fiber. So you have intermediate filaments. This is intermediate filament. This is inside the cell. It's from the inside the cell. Then we have microtubes. These are microtubes. These are microfilaments or actin filaments. You can also call it actin filament or microfilaments. This forms the cytoskeleton. The nucleus, the most important part in the cell. The nucleus houses the cell DNA in form of chromatin, directs the synthesis of ribosome and protein. So nucleus has a nuclear envelope. So it's a double structure membrane made up of phospholipid bilayer. So this is the nuclear envelope. This is double structure made of phospholipid. It has nuclear pore that helps the passage of iron, and molecules, iron molecules or RNA from the cytoplasm to the nucleoplasm. So nucleoplasm is the fluid within the nucleus. Chromatin that forms the chromosomes. Okay, the chromatin forms the chromosomes. It's made up of DNA and is the hereditary material. It's made of DNA and is a hereditary material. So like in human we have 46 chromosomes. In fruit fly there are 8 chromosomes. So that way the chromosome number changes with the organism. Nucleolus is the dark structure within the nucleus, and it's an aggregate of ribosomal RNA. It has ribosomal RNA, and proteins. So it forms a dark structure, and the outer part is connected to endoplasmic reticulum. So this forms the complete structure of a nucleus within the nuclear envelope. Endoplasmic reticulum. The endoplasmic reticulum is a series of interconnected membranes, tubes that collectively modify protein and synthesize lipids. So their main function is modifying proteins and synthesize lipid. It is two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. The rough endoplasmic reticulum is a network of continuous sacs. So it's a network of sacs. It is studied by ribosomes. These are the small ribosomes studied on endoplasmic reticulum. This forms the rough endoplasmic reticulum, and it forms the internal delivery system in the cell. So in the cell, suppose this is the cell and this is nucleus, then this forms the internal delivery system between the cell and the outer membrane. The next is a smooth endoplasmic reticulum. This is not having ribosomes. It is without ribosomes, and it synthesizes carbohydrates, lipids and steroid hormones. So no ribosomes embedded. And it also helps in detoxification of the cell, storage of calcium ion. These are small functions of smooth endoplasmic reticulum. Golgi bodies. The sorting, tagging, packing and distribution of lipids and protein takes place in Golgi apparatus. The protein packing plant modifies proteins and lipids made by endogenous reticulum and prepares them for export from the cell. Now, if you see here, these are Golgi bodies. Golgi bodies are flattened sac-like structure. It's a sac-like structure which has a curl endings we have. They have a role in plants, they have a role in synthesizing polysaccharides. In plants, they have additional role in synthesizing polysaccharides. So you have a cis phase, this is a receiving phase of the Golgi apparatus, and you have a trans phase. So that's the shipping, the releasing phase of the Golgi apparatus. So the material which is collected here, it is transported to the outer membrane with the help of transport vesicles or secretory vesicles. The secretory vesicles, they go and connect to the plasma membrane and take the material outside the cell. So this is how the Golgi bodies transport material from inside the cell to the outside of the cell. These secretory vesicles fuse with the plasma membrane and release the content outside the cell. So this goes and fuse with the plasma membrane and they release the material outside. This is how the transportation takes place. You can clearly see here, this is much more clearer. So you have a protein to be exported, transport vesicles, the cis phase, the receiving phase, and this comes from the endoplasmic reticulum and it moves, the transport vesicle or secretory vesicle gets connected to the plasma membrane and it is moved out. So this is the trans phase, releasing phase, and this is receiving phase. So two phases are there. Lysosomes. It's a digestive plant for protein, fats and carbohydrates. Digestive enzymes break down cellular waste and debris. It consists of enzymes. Transports undigested material to cell membrane for removal. So one of the good examples of lysosome functioning is the group of WBC, where you can see the macrophage. It's called a macrophage. It's part of the immune system. Whenever a pathogen enters the cell system, so it is engulfed. It's engulfed by plasma membrane, and the pathogen is pitched to the engulfed by the plasma membrane and forms a vesicles. And the vesicle move forward, it fuses with the lysosome and lysosome destroys the pathogen and it is destroyed. So this complete process is also called phagocytosis, where the pathogen is destroyed by lysosomes. And lysosome also, when it bursts, the cell itself gets destroyed. It has enzymes in it. Mitochondria. Mitochondria is often called as the powerhouse of energy factory of the cell because they are responsible for making adenosine triphosphate. That's the energy molecule, ATP, the cell's main energy carrying molecule. Mitochondria are oval shaped, double membranes organelles that have their own ribosome and DNA. The inner fold layer is called cistae. The area around the fold is called mitochondrial matrix. So you can see this inner membrane, outer membrane, cistae, the folding is called cistae, then the matrix. So these are the structure of mitochondria. Ribosomes, peroxisomes. Ribosomes is a cellular structure responsible for protein synthesis. Either the cluster or single tiny dots floats freely in the cytosome. So it's free floating in cytoplasm. Attached to to either the cytoplasmic side of the plasma membrane or cytoplasmic side of the endoplasmic reticulum. So either it's connected to rough endoplasmic reticulum or in the plasma membrane. Peroxisomes. Small and round organelles enclosed by single membrane. They carry out oxidation reaction that breaks down fatty acids and amino acids. They also detoxify many poisons that may enter the body. So this acts as a detoxifying agent in the cell and protects the cell. I'm going in brief all the structure so that we can cover maximum organelles in the eukaryotic cells. Vacuoles and vesicles. Vesicles and vacuoles are membrane bound sacs. They are membrane bound sacs that function in storage and transportation. Vacuoles are larger than vesicles and do not fuse with the plasma. This is one of the major difference. They do not fuse with the plasma membrane, but vesicles, they fuse with other membranes. Like we saw the secretory vesicles fusing, exporting material in the Golgi apparatus, fusing with the outer plasma membrane and taking the material outside, exporting the material outside the cell. So these vesicles fuses with the other membranes. Animal vacuoles, membrane bound sacs for storage, digestion and waste removal, and contains water solution. In case of plant, they have larger central vacuole that stores water, nutrients needed by the cell, helps to support the shape of the cell. So if there is a stress of water in the plant, the plant vacuole water is removed and the desiccation takes place, and you have seen the drooping of the plant takes place. So this is a plant vacuole. So this is a big area, big portion, it's a membrane covered sac having water, and this is the animal vacuole. Chloroplast. Chloroplast functions in the photosynthesis and found in plant and algae. It's not found in animal cells. In photosynthesis, the carbon dioxide, water, light energy are used to make glucose and oxygen. So the chloroplast, it has chlorophyll in it. It captures with the help of light and carbon dioxide. It undergoes photosynthesis, creates food for the plant. Now it has outer membrane, inner membrane, and it has structure called thylakoids. So thylakoids, the complete stack of thylakoid is called granum, and the matrix between the aqueous fluid within the thylakoids or the granum is called stroma. So this forms the complete structure of the chloroplast. Now this helps in photosynthesis in the plant cell. We have gone through the complete various organs in the plant and animal cell. Just to brief up, so these are the structures in the plant and animal cell: plasma membrane, cell wall, nuclear membrane, nucleus, centrosome, smooth and rough endoglazed reticulum, vacuole. So this vacuole is bigger in the plant cell, Golgi bodies, mitochondria, amyloplast, chloroplast. The same thing is in animal cell also. So these are the two structures we have just now studied. Coming to the GAN's video questions. What structures plant cell has and animal cell does not have? Where is the chromosome found in the cell? Difference between plant cell and animal cell wall. You can subscribe to QuickGlance video channel. Just click on the subscribe, and you can click on the notification icon also, so that in future uploads you always are updated by the various topics I am covering. Thank you.